Hello, everyone, and welcome to our session and an update on upcoming developments and investigational CFTR modulators and therapies that restore CFTR function. My name is Amit Euler, and I'm a MedPeds trained pulmonologist and director of the Adult CF program here at the Boston Children's Hospital and Brigham and Women's Hospital CF Center. I would like to thank Raksha Jain and Paul McNally and the NACFC conference organizers for the invitation to speak with you today. My disclosures include being the North American PI of the phase 2 studies involving both VX-121-101 and the ELOX-02, and I'm also the adult director of the local TDN. Many of us are fortunate enough to be aware of the impact of CFTR modulators and small molecule therapies and how they have transformed the lives and the management of people with CF. Currently approved, highly effective modulators are now available for about 90% of people with CF and include those with at least one F508del allele or now a CFTR mutation that is responsive based on in vitro data. However, we also recognize that CFTR function isn't fully restored and available to everyone with CF, so there's room for improvement. And despite these incredible advances, 10% of people with CF are not eligible to be on currently approved modulators, and other drugs that restore CFTR function are not yet available for others. For those who are fortunate enough to be on modulators like the highly effective Lexicaftor, Tazecaftor, and Ivacaftor, the majority of patients haven't fully achieved full restoration of their CFTR function, as evidenced by most with sweat chloride levels still above abnormal levels. And there's still room to optimize adherence and patient outcomes. Many of you are familiar with the drug development pipeline found on the CFF website, and as you can see, it is exciting to see the many sponsors introducing therapies targeting the basic defect in various stages of development. And here we see the potentiators and the combination potentiator correctors currently available to patients with eligible CFTR mutations. Here are several agents with either in phase 2 or just completed phase 2 clinical trials, including CFTR restoring potentiators and modulators from AbbVie, with clinical trials mostly being performed overseas. And ELOX-02, a small molecule therapy to restore CFTR, though not a potentiator or corrector, it is a compound that allows cells to override the premature and erroneous stop signal the mutation creates and to make a full-length wild-type or functional CFTR protein. And more on this in a minute. And then we have a phase 2 trial and soon to begin phase 3 trial that was finalized last year, a combination of two correctors, VX-121 and Tazecaftor, along with deuterated versions of Ivacaftor called VX-561 to allow once daily dosing, and more on this later as well. So I decided to include this slide, but won't spend much time on it since there's little publicly available information, studies being performed overseas, as I mentioned. Of note, ABBV2222, a corrector, did show a significant decrease in sweat chloride that was dose dependent, despite not showing a difference in lung function, though we wouldn't expect this in a short phase 2 trial with limited numbers. And currently underway is another phase 2 trial looking at the safety and effectiveness of ABBV3067, a potentiator alone and in combination with ABBV222. So more on that in the future. Back to ELOX-02, we've known for years about the ability of immunoglycoside antibiotics and similar molecules and their ability to induce conformational change and reduce the accuracy of codon, anti-codon recognition to increase the probability that translational read-through of stop codons occur. However, their impact will go away once treatment is discontinued, similar to current CFTR modulators, and prolonged sustained use lead not only to antibiotic drug resistance, but lead to intolerable adverse consequences to hearing and renal function. So as you can see here, ELOX is a eukaryotic ribosomal selective glycoside that was optimized with these chemical modifications to allow for more effective and continuous delivery. And the lower affinity of ELOX-02 for prokaryotic and mitochondrial ribosomes based on those chemical modifications is shown here and compared to gentamicin and G418, thus showing improved read-through without binding to bacterial ribosomes and optimized to bind eukaryotic ribosomes. And this leads to 100-fold lower antibacterial activity. And as we can see here, a 9-fold higher cytoplasmic ribosome read-through activity. Here we see the allelic frequency of common nonsense mutations, with G542X being the most common, followed by W1282X, R553X, R1162X, E60X, with 23% comprising the rest. In this study by Sermet Gaudelis, 6 of 9 patients with Y122X showed evidence of CFTR protein detection at the membrane of basal epithelial cells, an increase in CFTR-dependent chloride secretion with MPD measurements. Mean chloride concentrations decreased significantly and normalized in 2 patients, but those were those patients with CFTR protein expression. So very promising. In this slide, we can see internal data provided by ELOX showing CFTR-dependent forskolin-induced swelling in organoids from patients with differing nonsense mutations. CFTR-dependent swelling after 48 hours of incubation with ELOX-02 showed significant elevation of CFTR activity in organoids from all nonsense mutations, as you can see here. Those tested with G542X and R1162X, though, revealing the most activity. In our ongoing phase 2 ELOX-02 dose escalation study, for those with at least one G542X mutation and without F508del, or other mutation eligible for a modulator, there are 4 treatment periods of dose escalation, and subject recruitment is nearly complete. However, an amendment was just introduced and before the TDN Protocol Review Committee now and the FDA to add an additional treatment period 5 combining ELOX-02 with ivacaftor. Based on ELOX internal data and using organoid swelling assays, there was an increased activity with the addition of ivacaftor to ELOX-02 in those with CF patient-derived cells homozygous for G542X and W1282X homozygous patients, as you can see here, with incubation days 2, 4, and 6 shown. And similar results were seen using an Using chamber measuring short circuit current with ELOX-02 and escalating doses of ivacaftor, as you can see here. And this is a cells derived from G542X and an F508del patient. Thus, this amendment will increase the exposure of time to ELOX-02 compared to other treatments periods and allowing for longer-term safety and efficacy data and demonstrate in other CFTR modulator, as demonstrated in other CFTR modulator trials, and will include nonsense mutations beyond G542X and in combination with ivacaftor. More clearly shown in this graphic, there will be a 1 week of ELOX-02 monotherapy followed by 4 weeks of ELOX-02 and ivacaftor with additional nonsense mutations. And thus the endpoints will be sweat chloride and FEV1 as secondary endpoints, but primary endpoint will be safety and safety. Thus, in summary, ELOX-02 is an immunoglycoside designed as a read-through agent. It improves across CFTR nonsense mutations based on electrophysiology and organoid swelling. The clinical trials are ongoing to evaluate the efficacy of subcutaneous ELOX-02, that's how it is delivered, across doses, across nonsense mutations, and now alone and in combination with ivacaftor. Now shifting gears to rationale for a better combination potentiator corrector CFTR modulator in the era of elexacaftor, tezacaftor, and ivacaftor. Many of us have known that sweat chloride doesn't predict disease course in patients. In the study conducted by Fiedler and others, they determined that for any given study, reductions in sweat chloride levels and improvements in absolute FEV1 were not correlated for individual patients. However, correction of sweat chloride continues to be a meaningful outcome. And when the data from all studies were combined, a statistically significant correlation between sweat chloride and levels of predicted FEV1 changes were observed. Thus, sweat chloride level changes in response to potentiator of the CFTR protein by ivacaftor appear to be more predictive of pharmacodynamic biomarker of lung function changes on a population basis, though recognizing it is unsuitable for the prediction of treatment based on individuals. And we are looking forward to gaining more insight into this observation with the CHECK-SC study, or also known as the Characterizing CFTR Modulated Changes in Sweat Chloride and Their Association with Clinical Outcome Study. This is an epidemiologic study to determine the relationship between sweat chloride and response and clinical outcomes in individuals with CF on commercially available CFTR modulators. So we believe this makes the argument that sweat chloride is a meaningful outcome, though maybe not at the individual level, but at the population level for sure. In addition, and based on those studies looking at disease progression based on FEV1 and CF with data from US and UK patient registries, we know that the chronic decline in lung function is slowed, although not stopped in patients with Ivor Captain. And we believe we'll see similarly with ongoing studies looking at our triple combination modulators. Thus, more effective CFTR restoration may lead to to improve lung function stability and to reason to further investigate other combinations of modulators. In addition to that rationale, one more is to improve upon the current landscape of CFTR modulators, and it has to do with adherence. From a study by Price and others looking at once versus twice daily use of inhaled steroids in asthmatics, it favored once daily dosing as calculated by administered doses divided by scheduled doses as measured by both a dose counter and patient self-report. And in another study by Lalibert looking at the impact of once compared to twice daily dosing in a cohort of patients diagnosed with DVTs or venous thromboembolism, this multivariate logistic regression modeling probability of adherence to once daily to twice daily with a significantly higher odds ratio in favor of the once daily dosing group. In this study by our colleague Chris Siracusa and colleagues looked at Ivor Captain adherence rates, and despite the highly effective impact on the lives of patients receiving this therapy, adherence was variable. The figure on the left shows adherence using 3 different methods of reporting, including self-report, medication possession ratio, and electronic monitoring. And you can see the variable levels of adherence in the figure on the right, which depicts weekly adherence on the top graphic and dosing intervals on the bottom. And to cut to the chase, it shows adherence decreasing over time and dosing intervals increasing over time, showing once again the challenges beyond once daily dosing of medications with even the most celebrated of therapies. And there are many other studies duplicating this work with just not enough time to share it here, but you understand the points I'm making. So based on preclinical in vitro data showing improved chloride transport in human-derived bronchoepithelial cells compared to currently approved triple therapy, and as we know, these are highly predictive of clinical results in sweat chloride and clinical outcomes. As well as early phase 1-2 studies, Vertex introduced VX-121, tazecaftor, and VX-561, or deuterated ivacaftor, as 2 phase 2 studies looking at two-part phase 2 study looking at improving upon highly effective therapies. The study VX-121-101 Part 1 for those with one F508del allele and another minimal function mutation, as well as Part 2 for those with 2 copies of F508del and those already on tazecaftor ivacaftor. Many of you are already aware of these studies and hoping to publish our findings very soon, but I believe I make the argument that we need to improve on current modulators and to improve CFTR restoration based on improvement on adherence. A unique aspect of this study is the introduction also of VX-561, or deuterated isotope of ivacaftor, with a reduced rate of clearance, increased exposure, greater plasma levels at 24 hours, and a longer half-life compared to regular ivacaftor, and supporting once daily dosing of the combination of VX-121 tazecaftor and VX-561 together. On this Part 1 phase 2 randomized double-blind controlled study to evaluate the safety and efficacy of VX-121 combination therapy in subjects age 18 and older with CF and with 508del and a minimal function mutation, there was a significant increase in the absolute FEV1 and a corresponding significant impact on sweat chloride, as you can see here. And available from a press release on the Vertex website from July 28th of this year. A dose response was not observed, but this was expected with such small numbers. In the phase 2 study, while a dose response was observed with sweat chloride, however. And as you can see, sweat chloride, a decrease of minus 49.5 millimoles per liter. In Part 2, we looked at those homozygous for F508del with a tazecaftor ivacaftor run-in period. And here we once again can see a remarkable response in absolute FEV1 improvement up to 15.9% absolute improvement in FEV1 at day 29, and a corresponding significant improvement in sweat chloride of minus 45.5 millimoles per liter. And these results once again are available on the press release found on the Vertex website from July 28th of this year. Thus, this favorable benefit risk profile demonstrated in these studies, and also with the arguments that I made on adherence and further restoration and complete restoration of CFTR, supports further investigation of the combination of VX-121, tezacaftor, and deuterated ivacaftor in people with CF. And thus, phase 3 studies will soon begin and will evaluate the potential for the efficacy and safety beyond elexacaftor, tezacaftor, and ivacaftor. And we also look forward to sharing the complete data from our phase 2 studies in a manuscript coming very soon. So in conclusion, additional modulators will provide options for those who are less responsive or unable to tolerate commercially available modulators. Clinical trials are ongoing to evaluate the efficacy of subcutaneous ELOX-02 across doses, across nonsense mutations, and also alone in combination with ivacaftor, which is very exciting. And without fully normalizing sweat chloride levels, and we already know that elexacaftor, tezacaftor, and ivacaftor have not done that below the threshold of 30 millimoles per liter, there's room for more effective modulators and may impact long-term outcomes and further stabilize lung function decline. And also, once daily dosing will lead to better adherence and thus optimize CFTR restoration potential. And with that, I would like to thank the Brigham and Boston Children's Adult CF program and our incredible clinical research team, as well as the clinical research teams of participating sites, and foremost, our amazing patients. I'd like to thank those on the ELOX-012 study teams at National Jewish, Johns Hopkins, Nationwide Children's, Stanford, Baylor, Memorial Care Long Beach, and to the determined patients who want to help find therapeutic solutions for those without access to current CFTR restoring therapies, and to the VX-121-101 sites and investigators. We're all in this together, and we couldn't do it without each other. Thank you.
Hello, this is Dave Nichols. I'm presenting today on the topic of studying the benefits of modulator therapy in clinical care. And in many ways, this was an invitation to present the first phase PROMISE results. It's my privilege to do so. In way of disclosures, I acknowledge grants and also some consultation work I do as part of the TDN Coordinating Center. I want to start actually before getting into the results with acknowledgements of the many investigators who've come together for a couple of years or more to develop and conduct and now start to analyze the results of this study. They're all leading important sub-studies focused on different outcomes, and most of those have longer-term outcomes than this first phase or 6 months. So those results are to come, but we just really appreciate their input in these core results, and I want to acknowledge this great team that I've been privileged to work with, and especially Dr. Stephen Rowe, the overall study PI. Also, the TDN Coordinating Center and a critical group of people with whom this work would not be done. Specifically want to call out Alex Painter and others in the statistical team who are responsible for many of the figures we'll share with you today. We've organized this talk to present to you 10 important findings through the first 6 months of ETI use within clinical care in the US. To remind those less familiar, this is the phase 1 portion of PROMISE. This was pre-planned to look at and actually publish results from the first 6 months, and this mirrors in some ways the randomized controlled trials that were done, allowing for some comparison there. The study is ongoing, and there will be longer-term outcome measures at 18 and 30 months, perhaps even longer. So who did we enroll? Overall, we enrolled about 500 individuals with CF. You can see there in the red box. And then to the right of that, they're categorized based on the modulator they were using at their baseline visit before starting ETI, or elexacaftor, tezacaftor, ivacaftor. So those coming in on no modulator were, for the most part, heterozygote for F508del. Those coming in on ivacaftor had a G551D/F508 mutation. All but one of them did. And then those coming in on 2-drug therapy were our homozygote population. In way of demographics, 25 years old on average, baseline lung function about 80% of predicted, BMI right at 23, adolescents age 12 to 18, 56 percentile for BMI. About half of the group coming in were homozygous for F508del, and about another 40% heterozygous for F508del with a non-G551D mutation. So number one, the point we'd like to make is that ETI is the most effective drug therapy ever studied in an observational CF study in the US like this. To try to convey that point, we'll show you these data. So they're organized by color. Blue will be those who are on no prior modulator at the start of the PROMISE study. Those who are on a 2-drug modulator combination are in green, and then those coming in Ivor Captain are in orange. Looking at lung function, absolute change in percent predicted FEV1, you can see coming from baseline to 6 months, very nice improvements in all of these groups, 9 to close to 11% predicted in the first 2 groups, and those coming in already on highly effective therapy with Ivor Captain with a 6% further improvement. Change in sweat chloride, similarly impressive, greater than 40-point reduction toward normal status in the first 2 groups, nearly 25-point reduction in those coming in an Ivor Captain. And CFTR respiratory domain, or signs and symptoms of a reported pulmonary disease in CF, dramatic improvements in all 3 groups. So to put this in a little bit of context for comparison, the GOAL study, looking at those with G551D mutation who were starting modulator therapy for the first time, in that case Ivor Captain, had just under 7% improvement in FEV1, sweat chloride drop of nearly 54 millimoles per liter, and a respiratory domain score that increased by about 7 points at 6 months. Number 2, drug intolerance and discontinuations have been low, and adherence appears to be high in the PROMISE study to date. This is a little bit indirect, but the way that we get at that is this. By self-reported usage at 1 month, 99% of individuals completing that study were reporting using ETI, and at 6 months, that had changed very little. And we have retained pretty well in the study. Over 96% of people who started ETI remain in our PROMISE study with us now. Another way to look at this, more so adherence, might be change in sweat chloride as a biomarker of drug use. And we see that from the 1 month to now the 6 month time point, sweat chloride has been remarkably stable in these groups. Number 3, newly established baseline health status after ETI is remarkably good. I think this is really encouraging, not only for people with CF and their families, but for those of us who provide care for them. So if we look not at the change, but rather where they start and end in these outcome measures, you can see that these groups reach an FEV1 at or near or just above 90% of predicted, which is terrific. Sweat chloride are all below, on average, 60 millimoles per liter. The homozygote population reached near 40 millimoles per liter, and the Ivor Captain responsive group, the G551D group for the most part, nearly 25 millimoles per liter. The respiratory domain scores similarly are impressive in that on a scale of 0 to 100, they're reporting on average scores of 90. So very few signs or symptoms of respiratory illness over the last 2 weeks. And BMI, similarly impressive in that the BMI in adults reached 24.5 and the Z-score BMI in children plus 0.5 on average. So an average study participant in PROMISE at 6 months into drug therapy is about 25 years old with lung function of 90% of predicted, sweat chloride below 60 millimoles per liter, BMI nearing overweight categories, and reporting almost no respiratory symptoms in the last 2 weeks. Number 4, people already using the prior benchmark for effective modulator drug therapy see impressive further gains when transitioning to ETI to target the F508 allele that they have. So that group, a smaller group, came in a little older, 27 years old on average, FEV1 79% of predicted, BMI of 23. They had a greater than 6% further improvement in their lung function. Sweat chloride decreased by 24 millimoles per liter, BMI increased by 1.2 kilograms per meter squared, and their respiratory domain scores went up by over 18 points, which I think is very impressive. Number 5, 2 drug combinations have had sustained biological effect, and having 2 alleles to target with ETI appears to be more responsive than having 1. Again, these data are a little bit indirect, but the way that we get at that is this. Those who came in on 2-drug therapy, the homozygote population, started about 10 points lower or more normal in their sweat chloride compared with the heterozygote group, but they had the same overall response in transitioning or starting ETI. And so they end up about 10 points lower versus the F508del heterozygote group coming in on no modulator therapy. Another way to look at this would be to go back to the registry data and get their diagnostic sweat chlorides, which was done. So you can see on the far left, when they're on no modulator, their mean value is 102 millimoles per liter. At the beginning of PROMISE, when they're on 2-drug therapy, it's 85, so 15 points or so lower, suggesting sustained effect of that drug therapy. And then you can see much greater improvements at 1 and 6 months into PROMISE study showing the 3-drug combination in blue. For comparison, we see from a no modulator status or diagnostic sweat to 3-drug combination, a 61 millimole per liter change. And in reference or comparison with goal, the change at 6 months was 54 millimoles per liter. Number 6, we're actually starting to have to think about ceiling effects in some of our outcome measures in clinical trials in populations like this. Here we have our outcome measures that we focused on today, and you can see sweat chloride in particular for that Ivor Captain responsive group down in the 20 to 30 range. But even for the larger homozygote population, they're just over 40. FEV1, we see averages of near 90% predicted or even a little higher than that. This is a group, remember, that is in their mid-20s on average, has had a couple of decades of CF lung disease developed with modest or no CFTR modulator treatment. And their respiratory domains, perhaps more impressively, show values that are approaching that perfect score of 100, indicating no signs or symptoms. To dig into that a little bit more, we'll show you the data another way. So here are their CFTR respiratory domain scores. At the top in light green is a maximum score, no signs or symptoms reported. And then just below that in the teal color is a good score of 80 or higher. You can see at baseline only 4% of people reported a perfect score, but that increased to 35% by 6 months. And overall, 85% of people were reporting a score of 80 or higher. This broke down actually to be present even in those who started with baseline lung function, percent predicted FEV1 less than 65 at the top, where you can see by 6 months, a quarter of them are reporting perfect scores and a sizable number higher than 80. You can see that the trend actually increased as baseline lung function increased in the group overall. Average BMI is nearly in the overweight category. For adults, 24.5 for a mean, and in the adolescents, a z-score of plus 0.5. Again, working toward a z-score of 1 or more, which would be indicative of overweight categorization. Number 7, sweat chloride change was larger for female than male participants in the study. And their baseline values were pretty similar, as you can see there on the left column, but the change was 10 points greater in those female rather than male. There was a difference actually seen in CFQR respiratory domain improvement, where females had a greater improvement as well. Change in FEV1 did not correlate with sex or age in these groups. This larger sweat chloride response or change in females was seen previously and reported with the Ivor Captain study and actually a study with 2-drug modulator therapy separately. It was thought that perhaps body weight differences could explain this, but some nice work done by our stats team, led by Alex, suggests that weight is not fully explanatory, at least in the PROMISE cohort. And so to my knowledge, the full biological explanation for this is yet to be fully understood. Number 8, mutations that have been added to the FDA approval list for ETI based largely on in vitro testing appear to have larger sweat chloride response compared with those who would have a mutation not expected to respond to ETI. Now, all of these individuals have an F508del. This is a heterozygote group. And then there were 11 people in that group who on the other allele had 9 of these mutations on the approved list. They're shown there at the bottom for your reference. We compared them with 45 other people who had a mutation that was not believed to respond to ETI. And you can see that those on the approved list had a 13.4 millimole per liter greater change or reduction in sweat chloride compared with the non-responsive group. Number 9, for what appears to be the first time, we see correlation between sweat chloride change and change in percent predicted FEV1 in a single study of this duration, 6 months. And so the correlation perhaps could be stronger. It's 0.19, but it was highly statistically significant. And I think it's nice to see a connection here between a biological response in sweat chloride and a fairly complicated measurement of pulmonary status with FEV1. Number 10, restricting PROMIS by some key eligibility criteria for the randomized controlled trials actually produces very similar responses to ETI in this real-world observation. And so that suggests that in clinical use, these drugs may be working about as well as they did in very impressive results from those clinical trials. Here I'll show you actually some 1-month changes to better align with some of the reports in those clinical trials across the top. FEV1 on the left, sweat chloride in the middle, respiratory domain changes on the right. And so we have the 2 trials to compare with, the homozygote study, the randomized controlled trial in the middle there on the left, and then the heterozygote study there on the lower left. If we look at FEV1, when we restrict PROMIS to that group and actually remove quite a large number of people from PROMIS, you see that the outcomes overlap very closely in regards to FEV1, and they're even closer in regards to sweat chloride, where we're really within 1 or 2 millimoles per liter change. And the CFTR respiratory domain changes are basically on top of one another. All right, so that's 10 interesting, perhaps important findings. And I want to also highlight some of the sub-studies that are beginning to present data as well. And as I said earlier, more to come. First of all, airway microbiology. This has not been a big focus of studies in regards to the randomized controlled trials. And so I just want to highlight that one here. It's led by Dr. Singh for the most part, also Dr. Hoffman and his National Resource Center lab, and I've been involved in some of this too. Privileged to work with these guys. So reporting out the sputum density, CFUs per mil, all of our work here is in sputum. And we have Staph aureus on the left, Pseudomonas aeruginosa in the middle, Stenotrophomonas on the right. 2 to 3 log or even larger reduction, and that's been sustained from 1 month on to 6 months. These are averages among the group of people with that particular pathogen at baseline before starting ETI. I think that's very encouraging. That's hundreds of fold reduction, and it's exciting to see that, particularly in a group this old. Note that in the Pseudomonas group and the Stenotrophomonas group, fairly large numbers go from culture positive at 0 there before drug to culture negative at 1 month. And many of those have stayed in that category. I'll show you that a little more. So this is the percentage of people who were culture negative for their last 2 visits. They provided samples, sputum samples. Both of those cultures were negative. And in this group, these were individuals who were culture positive at their baseline visit before ETI. So over 30% in Pseudomonas, over 60% in Stenotrophomonas. But there is still more to learn, and work continues to better understand this. If we take those same samples and apply rigorous additional or repeat culture, a few of the PA negative individuals are lost. This focuses on Pseudomonas here on the right. And then if you take those samples and you look carefully with digital drop PCR techniques, over half of those who appeared to be culture negative at first do appear to have some evidence of Pseudomonas present in those samples. There's more to learn there for sure. PCR can detect both live and dead bacteria. What does it mean to have very low concentrations of a pathogen in the airway? Is that clinically relevant? I think important questions to consider in the future. I want to point everyone toward the workshop in 04. Sarah Morgan from the Sing Lab will be presenting more details on all of this and can answer more of your questions. She'll also be showing the early microbiome findings. The Mucociliary Clearance and Sputum Rheology study looked at baseline and then 1 month into ETI drug therapy, showed very nice improvements in mucociliary clearance from the lungs. You can see baseline in gray and then improved clearance in blue across those time points. And the character of the sputum also improved. So reduced percent solids in the sputum and reduced viscosity as well, statistically significant on both accounts. They also use their micro OCT imaging technique to look at the nasal airway epithelia and to identify improved mucociliary transport in a relatively small number of people, but still reaching statistical significance, which is exciting. And I want to credit the investigators below. The mucociliary study and the mucus biology study is led by Drs. Donaldson and Rowe. And then some other key findings are beginning to emerge in the GI group led by Dr. Schwarzenberg and Friedman have shared with us some high-level results. They've seen relatively small changes in GI symptom scores across a number of PROs in the group, and the fecal elastase 1 measurements of innate pancreatic function do not appear to have improved over the first 6 months of ETI therapy in this group age 12 and older. I encourage you to visit their poster and to reach out to the lead GI investigators for more details. Lastly, just want to highlight 6 papers that have been published already this year, looking at other outcome measures in real-world use of highly effective modulator drug ETI. And this is not an exhaustive list, but it's great to see people looking at outcomes that are not included in PROMISE, and you can see those highlighted in yellow. And lastly, I want to thank the 56 sites who are participating in this study. It was not the easiest of all times to do a study like this, requiring some in-person visits. The way they've rallied to do this as well as they have is really remarkable and to their credit. Not to mention the people with CF and their families, without whom none of this would happen, and for whom we hope that these results are encouraging. So thank you very much for the chance to present on behalf of the PROMISE team, and we look forward to the Q&A time later.
Hello, everyone. My name is Herman Bieler, and I work at the CF Foundation's laboratory in Lexington, near Boston. Together with my colleague, JP Clancy, we are excited to update you on our work to expand highly effective CFTR modulator therapy coverage via precision medicine. You'll first hear about theratyping in FRT cells, followed by a discussion of personalized testing with patient-derived nasal cells. I have nothing to disclose. There might be many ways to describe theratyping, but you might want to think of it as matching therapeutics to genotypes. There are over 1,000 variants of the CFTR gene that have been associated with CF, and the majority of those are found in 5 or fewer people. This makes traditional clinical trial designs and drug approval for these rare variants all but impossible. Now, in 2017, the FDA opened the door to the use of established in vitro assays to assess the responsiveness of pathogenic mutations to CFTR modulators, and thus allowed label expansion for these groundbreaking therapies based on in vitro data. The Foundation took this opportunity to sponsor a large-scale theratyping effort that has been conducted at the labs of Dr. Bob Bridges, Dr. Phil Thomas, and the CFFT lab headed by Dr. Martin Menze. Each lab screened over 650 CFTR variants in the Fisher-Rat thyroid or short FRT cell line for their responsiveness to lumacaftor ivacaftor and elexacaftor tezacaftor ivacaftor. As you know, on December 21st of last year, the FDA expanded its approval of 3 CFTR modulator therapies to include over 600 people with CF, or 2% of people with CF in the US who have certain rare mutations. Our goal is to identify many more variants via theratyping to basically enable modulator therapy for all responsive CFTR variants, beyond the currently approved 183 variants. Unfortunately, the FRT cell system does not allow the study of certain mutations. Gary Cutting from Johns Hopkins, who leads the CFTR2 team, put together a list of 650 plus variants suggested to be disease-causing that are potential targets for modulators. These are exonic variants reported in the CFTR2 database and expected to make CFTR protein. The majority of those are missense mutations, but we also included some indels and complex alleles. Now, intronic splice, PDC, or promoter variants are not included in our screening since we use a cDNA-based cell model. So we conducted 2 consecutive studies, a pilot screen, also called a double, where we tested variant responses to CFTR corrector lumacaftor and CFTR potentiator Ivor Captain in single and in combination, and a second screen, also called a triple, where we tested the variant responses to the corrector combo elexacaftor, tezacaftor, plus minus Ivor Captain. This slide describes the functional assay. On day 0, FRT cells which do not express endogenous CFTR are transfected with variant CFTR and seeded onto transwell filters. After 4 days on filter, cells are treated for 24 hours with DMSO or corrector compound. Potentiator VX770 or Ivor Captain is added during the functional assay on day 5. This graph shows the tech platform, which basically is a modified higher throughput Using chamber system that allows us to do parallel recordings from multiple 24-well transwell filter support plates. One of the wells is shown here. Now, this configuration allows us to measure CFTR function as CFTR-mediated transepithelial chloride conductance. And the formula here shows the relationship between conductance GT, the number of CFTR channels N, and CFTR ion channel open probability PO. So CFTR variants that respond to correctors or potentiators by an increase in N or PO or both will yield an increased transepithelial conductance upon activation of CFTR. To explain this in a bit more detail, this figure shows conductance traces of a responsive variants after 24-hour treatment with DMSO in white or corrector in yellow. After recording of the baseline conductance, forskolin is added to stimulate CFTR activity, followed by addition of Ivor Captain to test the variant response to Ivor Captain in the presence and absence of corrector compound. Addition of CFTR-specific inhibitor completes the assay. Now, the assay is designed to give individual and combined readouts for corrector and potentiator responses. And going forward, I will refer to the CFTR forskolin response in the absence of modulator treatment as CFTR or variant baseline activity. Variant baseline activities and variant modulator responses will be presented as % of wild-type forskolin response or % normal. This figure shows the plate layout of a 24-well assay plate. On each assay plate, we measured wild-type, G551D, and F508del controls, and 4 CFTR variants. F508del and variants are treated for 24 hours with DMSO or corrector, and wild-type G551D with DMSO only. Treatments are done in duplicates, which gives an N of 6 for each variant in treatment when combining data from all 3 sites. And thanks to Linda Millen from Phil Thomas Lab, we also have CFTR protein expression and maturation data from nearly every well of the assay plates that Linda ran in both studies. This slide shows the impressive cross-site concordance of functional readouts from the triple study. The graphs compare the average data as % of wild type from each site for over 650 variants. A correlation coefficient of 0.8, 0.9, I guess, speaks for itself. There is excellent correlation between all sites for both variant baseline activities as shown in the top panel and variant response to the triple as shown in the bottom panel. Now, this shows a representative subset of variants from the triple study, sorted by their functional response to forskolin. All data are normalized to wild type, equivalent to 100% on the y-axis. Benchmark levels for G551D plus Ivor Captain and for F508del plus triple and lumacaftor Ivor Captain are also indicated. The majority of variants exhibit little to modest baseline activity and are likely disease-causing mutations, but there is also a good number of suggested CF-associated variants that obviously don't show disease liability. The majority of those have an allele count of 1, and it is possible that the actual disease-causing mutation may have been missed in the genotyping process. Some of these variants, however, may actually be disease-causing, but their pathogenicity is not captured in our CFTR cDNA-based assay, for example, variants that disrupt splicing or bicarbonate transport. Now, as you can see, the majority of variants respond to modulator treatment with an increase in chloride transport activity. Vertex interprets an in vitro improvement of 10% of wild type, regardless of baseline function, likely to confer a clinical benefit at the population level. Variants that respond to modulated treatment by an increase of 10% of wild-type function or more are thus classified as responders. And by applying this response threshold to our data, we are in excellent agreement with Vertex. 95% of Trikafta-approved variants respond to modulated treatment by an increase of 10% of wild-type function or more in our hands. So let's have a look at the complete data sets from 650 plus variants, beginning with a double. Every black dot represents the baseline activity of a specific variant, and the red dots show the corresponding responsiveness of a variant to Lumacaftor, Ivor Captain. Black dots in the gray box represent variants with baseline activities of less than 10% of wild type or normal, and among those are variants that respond to combined modulated treatment by an increase of 10% of normal or more. Those are indicated by the red dots in the black box. So there are over 100 CFTR variants with a baseline activity of less than 10% of normal that respond to Lumacaftor, Ivor Captain by an increase in CFTR function of 10% of wild type or more. Now, this graph shows the same thing for the triple study. There are 230 CFTR variants with a baseline activity of less than 10% wild type function that respond to the triple combo by an increase in CFTR function of 10% of wild type or more. Removing all the Trikafta approved variants from the plot leaves us with an impressive number of 150 responsive CFTR variants with a baseline activity of less than 10% wild type function. Now, this is another way to summarize our functional data. Many more variants respond to the triple combination compared to Lumacaftor, Ivor Captain. In fact, 68% of variants with less than 10% normal function show significant improvement with a triple combo compared to 37% that respond to Lumacaftor, Ivor Captain. And at this time, we are in the process of finalizing the data set for peer review and publication. Also, please note that we are happy to provide variant plasmids to CF researchers for academic research as well as drug discovery or development. And while I had the honor of presenting this project, it can't be emphasized enough that this is a team effort between UT Southwestern, Rosalind Franklin University, Johns Hopkins, and the CFFT lab. And I want to thank everyone on the theratyping team for their contributions, especially Phil Thomas, who has been spearheading the project. And I want to thank you for your attention. And with that, JP, it is now your turn.
I'd like to thank the organizers for the opportunity to speak today at this session, talking about expanding highly effective modulator therapy coverage via personalized medicine. And again, my name is JP Clancy, Vice President of Clinical Research for the CF Foundation. Just quickly, my disclosures, there are none. And first thing I wanted to talk about is the difference between precision and versus personalized medicine. I think you'll see from Dr. Beeler's talk that precision really, when discussing bringing modulators to new communities, new populations with rare mutations, precision medicine is the type of testing that is being done in a heterologous expression system, looking at on a genotype specific way. While personalized medicine really implies doing testing on cells derived from a given person. And while there will be obviously the impact of modulators on the specific alleles, both of them in theory, there are also the biologic factors specific to that person that are incorporated into looking at the data and interpreting. So while they are related, they are somewhat different. And again, I'll be speaking about the personalized component of theratyping and testing. We'll go through what our CFF supported national resource centers are in terms of this availability for the CF research and care community, the use of model systems and specifically nasal epithelial cells, and then just the impact of having the center has been allowing patients to get access to modulators based on these results. So the National Research Centers were established roughly 20 years ago, and they're really focused on providing study sponsors and investigators centralized expertise in outcome measures relevant to conducting clinical research. And you can see this kind of relationship that currently exists. The TDN Coordinating Center, which helps to organize and to conduct clinical trials through the TDN network sites. The CFF, which provides support to the TDN Coordinating Center and through them to the different TDN sites. And also the National Resource Centers, which again, supported by the CF Foundation, work often with TDN Coordinating Center, but it also can work with investigators outside of the TDN. And I just highlight that this is a resource available to clinical researchers beyond the TDN confines. So in 2020, we had a RFA and funding cycle for National Resource Centers, and I won't go into all the details, but there are basically outcome measure advancement corps, which are shown on the left, and outcome measure service awards, and those are on the right. And the outcome measure advancements are basically those types of centers that perhaps are not quite as mature and are not perhaps ready for participation or inclusion in clinical trials at this time, but with some work may be ready to do that. While on the other side, the outcome measure service awards are really those that are much more mature and capable of being incorporated into clinical trials. A good example is the sweat analysis core, the Mucociliary Clearance core, multiple breath washout core, and so forth. So, but what we'll be talking about today is really focused on this nasal cell theratyping core up here at the top left. So this center is based in Cincinnati, and I actually helped establish it before the NRC RFA happened while I was still working there. And basically the idea is that brushed nasal cells are expanded and grown in conditions to allow them to de-differentiate somewhat and be able to increase the mass and the volume of cells available for testing. And then they're kind of grown in two separate sort of platforms. One is planar cultures, which they're at an air-liquid interface, that's shown on the top left. I'm sorry, and then down below are 3D spheroids, which are grown in matrigel. And they are really, they do measure related but independent things. The planar cultures help us look at CFTR-dependent ion transport, while the spheroids help us look at actual CFTR fluid transport. So the two are, again, related, but not exactly the same. So just to give you a quick comparison of nasal cells and bronchial cells, which are really probably what I would consider the gold standard for studying CFTR modulators outside of patients in clinical trials. Dr. John Brewington and colleagues performed a study a couple of years ago looking at sort of the performance of nasal cells and bronchial cells obtained from the same person at the same time. And basically, they did a simultaneous nasal brushing and bronchial brushing during a bronchoscopy that was scheduled for clinical care. This included a total of 10 subjects. You can see their age, roughly 10 years. Sweat chloride was around 91. And mean lung function was around 86%. And again, these were patients, by and large, who either had very classic CF or perhaps variants that were still being defined. And so there were variable genotypes within this group. But what we're looking at on the right side are a couple of the endpoints of many that were in this paper. One is that the cells grow in a very similar fashion as we look at the HNEs in gray circles and the HBEs in the open circles and dotted lines. You can see that their growth over time and over passages is very similar. And then when we look over here on the right side, we're looking at nasal epithelial cells and bronchial cells, looking at their CFTR function. And this is stimulated, the first bar is cyclic AMP stimulation, and then the small bar is the addition of Ivor Captain or VX770. And then the bottom is just the total inhibited CFTR-dependent current. And you can see that by and large, they're rather similar, not exactly the same, but certainly not statistically significantly different. And so providing some reassurance that the model systems should perform in a similar fashion. So this is just a picture of the team. Dr. Brewington, there on the left, he is the director of the CCHMC Theratyping Advancement Corps. And then members of the team on the right, and these include Rory O'Shaughnessy, Hunter Morgan, Alicia Ostman, and Jessica Meeker. And I show you these names and faces because if you do need to contact them, Rory is probably the most appropriate person, simply because she provides the consenting in the kits, and her email address is available in our National Resource Center on the CFF Research tab website. So this is just a snapshot of the number of places that have sent nasal cells for testing, and you can see it's quite extensive. And again, this is a service that the the Theratyping Center has been providing for a number of years, but it's continued to evolve as modulators have evolved. But just to give you some general statistics, there's been a total of approximately 300 samples, or a little over that actually, that have been studied. And this has been actually in the context of both theratyping for modulator access, but also part of other studies like GOAL, in which nasal cells were obtained from patients, expanded, and then banked. So that's where that experience comes from. But when we look just at the theratyping, about 120 subjects or a little over have been enrolled. They've generally had a CF or had a suspected diagnosis. The genotype criteria for testing has varied, but evolves with the modulator as modulators become available to patients. But currently, the testing is really focused on those who are not candidates for CFTR modulators based on the current label, and that can be based on either actual phase 3 clinical trials, or it can be based on in vitro theratyping done as part of precision medicine that has allowed label expansion for those with very rare and ultra rare CFTR mutants. So those that have not been on that added to the label and do make some protein, and specifically modulators can only work if there is available CFTR protein. And if we look on the right side, we're looking at just the general contamination rate, and this is looking at either specimens obtained locally or obtained from outside centers. And the point is they're very similar, about 20% risk of having contamination occur. And so it's important to inform families that this is a possibility. It may need to be rebrushed. And the other thing is exciting is about drug access. What we're looking at on the bottom left is all just a kind of a pie chart looking at all the subjects who've undergone theratyping. The purple are those who are not responsive to CFTR modulators, while the green are those who are responsive and have gained access, and the orange are those whose cells have responded, but they have not been able to gain access. And this access, again, is through conversations with third-party payers, because this does represent off-label use of the modulators. And on the far right, you can see the different types of modulators that people have gained access to. Again, as time has evolved, and modulators have evolved, different type of testing has been conducted. So just to summarize, the H&E Theratyping Advancement Cohort at Cincinnati Children's is supported by the CF Foundation, and it does study patient-derived nasal cells. Focuses on those who currently are not candidates for modulators, who have mutations which are potentially responsive. And really what that means is there is some protein. It doesn't cost anything to send these samples and have them tested. Results are shared back to the referring physician, and also with an accompanying letter that describes the findings and what recommendations there are. And this has been often successful in conversations with third-party payers to obtain trials of modulators for these patients. And finally, the need for testing may decrease with ongoing label expansion and or as new modulators become available. So again, I would just like to say thank you. I very much appreciate the opportunity to present this work, and look forward to having a further conversation at our later in the session.
Hello, I'm Jane Davis, and it's my pleasure to talk to you today about the progress we've made with CFTR modulators into childhood and infancy, and to try and raise the question, can we prevent CF disease manifestations altogether by treating very early in life? Here are my disclosures. So I started by asking myself whether prevention of disease manifestations is a very big ask, or whether it's actually quite likely based on what we already know. And I think there's something to be said for both of those. CF babies at birth largely have normal lungs and lower airways, but we know that early structural changes do occur, and large animal models would suggest that newborns are not completely disease-free, demonstrating gas trapping and mucus gland obstruction. However, we know that those infants and children who remain infection-free have substantial health benefits. Conversely, the pancreas is diseased in the vast majority of newborns, and of course, a raised ILT forms the basis of most newborn screening programs. We've learned over the last few years that pancreatic insufficiency is not as irreversible as we had all thought. Of course, most other complications are either absent in the newborn period and arise later, So what do we know so far about the lungs and the ability of children to respond to highly effective modulator therapy? In cohorts aged 6 to 11 in clinical trials, their pulmonary efficacy has actually been very similar in terms of FEV1 to what's been seen in older groups. And that's despite them starting these trials with much less severe baseline lung disease. So you can see here graphs from Ivor Captain, Ivor on the right, and then triple therapy with Elextezova in the bottom left, and changes which really reflect the sort of changes which were seen in older, more severe populations. However, FEV1 may not be ideal in this age group and certainly not below this age group. And as an example, I show you here outcomes from lung clearance index from the same data set as shown in the Ivor FEV1 above. And you can see a much clearer distinction of the placebo and treatment groups with this more sensitive outcome measure. This is being increasingly used in clinical trials in early life and in milder disease, and I think will be more and more needed as we progress into the highly effective modulator era. For those of you who are interested in hearing more about lung clearance index, Chris Short, one of my PhD students who works with me on the LCI core facility, is presenting at Symposium 21 tomorrow. What do we know about lung infection and exacerbations that we could use as predictions for what might happen in this cohort? Most of the data we have at the moment is from older cohorts, but even here, reductions in chronic infection frequency seem to be documented, and we know that chronic infection is the most challenging. You can see here UK registry data looking at patients in the 4 years after they commenced on Ivor Captain, showing a decrease in the proportions of those recorded as having chronic Pseudomonas infection, whereas the comparator group remained stable. We mostly appreciate that eradication of early infection is easier than a clearance of chronic infection. So it is possible or even likely that early modulator treatment will delay or prevent chronic infection, but we really need data. We probably mostly need observational real-world studies which can be sufficiently long and large, linking in with registry studies to try and really answer these questions. The caveat to this is that we will have sampling challenges increasingly. Many people taking triple therapy become non-productive of sputum, and we therefore have to rely on slightly less sensitive and specific methods of assessing their lower airway microbiology. Also, in terms of pulmonary exacerbations, the frequency is reduced in older cohorts and trials, but not down to zero. This might be different in early childhood if these drugs are commenced very early, but we do have challenges defining pulmonary exacerbation in this age group, and these challenges may become even more complex. Moving away from the lung to other organs, we have seen ably demonstrated in multiple trials now that exocrine pancreatic function can be restored by modulators initiated early in life. This is seen very frequently in cohorts of young children, but has even been reported in older people. So on the left-hand side of the slide here, from the Arrival trial, this cohort aged 12 to 24 months, with the majority of the group experiencing levels of fecal elastase above the 200 microgram per gram cutoff, which indicates pancreatic exocrine insufficiency by the end of the 24-week period. As I said, though, this has also been reported in older patients, and here's a case series from one of our clinical fellows at Brompton, reporting on a number of patients experiencing restoration of pancreatic sufficiency after the age of 6 years. This is something that we need to learn more about, and we need to increasingly monitor in patients who have benefits from triple therapy. So can starting immediately at birth and giving as much functional restoration of CFTR as possible, either with current or future drugs, prevent pancreatic insufficiency developing in all patients? I think this remains to be seen, and we do need to continue to collect data. In terms of the endocrine pancreas, CF-related diabetes is in general a later complication of CF, but its frequency has been shown to be reduced even when highly effective modulators were started later in life. So I think we have good reason to expect a very substantial reduction in risk when these drugs are initiated early in life or even in infancy. But we urgently need to know more. We will continue to learn more from the clinical trials which are running, and I'm very pleased at the progress these have made into the pediatric space. The first of these drugs to come through the trials pipeline, of course, was Ivor Captain, used as a monotherapy. The license now extends down to 4 months of age in many parts of the world, and the Arrival trial is currently recruiting infants even younger than this. In terms of Elextez Ivor, license age is different in the US and Europe. We hope in Europe that this extension down to 6 to 11 will happen fairly soon. We do have trial data in that age group, the single-arm data that I've shown you, which is published, and we're also awaiting publication of a placebo-controlled double-blind trial in the near future. There has been a trial recently started in 2 to 5-year-olds, so we can imagine this reading out within the relatively near future. However, we need to bear in mind that clinical trials recruit a very defined subset of patients. They perform a limited number of outcome measures, and they are usually for a relatively short period of time. And most of the studies of these drugs have been designed with open label arms, largely designed to collect safety, but also maintenance of efficacy. And these are extremely valuable when we're looking at the long-term benefits of the drugs. But we will also learn a huge amount from the multiple real-world studies that are taking place worldwide. So the PROMISE study in North America, a prospective study to evaluate biological and clinical effects of significantly corrected CFTR function, has recruited a large number of patients on triple therapy, and there will be posters and abstracts reporting some of those data at this conference. In Ireland and in UK, Paul McNally and I are leading on the RECOVER study, which is somewhat similar in terms of its aims, funded by the CF Foundation, CF Ireland, and the CF Trust. We're recruiting patients commencing triple therapy and performing a large number of outcome measures on them, both those in the clinic, but also a large number respiratory and other organs over a 2-year period of time and hopefully beyond. Importantly, we're also using this platform to examine the impact of starting triple therapy on adherence and uptake of conventional medications. And coming to the younger age group, BEGIN is running in the US, and Paul McNally is just putting some flesh on the bones of a plan that we have to run a similar sort of study called ENHANCE, which establishes the natural history in an advanced new CF care era. So much younger children running alongside data from CF registries will be recruited to collect annual data of bespoke and advanced outcomes. And we will use this, we hope, as a platform to run individual real-world studies of varying intensity and duration, which can be bolted on. So how could the future look for today or tomorrow's newborns with CF? Modulators, which can lead to high-level CFTR function, given at the time of diagnosis, which can be within weeks of birth in areas with newborn screening, could lead to delayed onset or complete absence of pancreatic insufficiency. This may only be for a proportion of patients. I would hope that it would lead to sufficient mucociliary clearance to render airway clearance and physiotherapy unnecessary, and this would have a very substantial impact on the burden of care for families of these newborns. It may also mean that we don't need to routinely initiate hypertonic saline and DNAs in the way that we currently do. One could imagine that encountered infections would be cleared rapidly rather than taking foothold in the lung, and that chronic infection would become rare. And I would hope that structural and functional lung health will be well maintained. If this were to happen, we need to give some careful thought to how we best deliver CF clinical care. The rule book probably needs a rewrite. We may find that hospital attendance can be less frequent or regular and rather tailored to individual patient need. We can build on the huge experience that has been catalyzed by the COVID-19 pandemic in home and digital monitoring. However, I think it's very important to remember that these children will need sensitive measures to monitor for their lung disease, which may otherwise remain silent and give us a falsely reassuring picture. So measures such as multi-breath washout and lung clearance index or imaging will require patients to be present in hospital. Defining pulmonary exacerbations, as I've said, is already tricky in this age group and may need adapting further, but I would envisage that hospital numbers will likely be lower. And hopefully, for a majority, at least in the early years, nutritional management could be more like that of a normal child rather than a CF child who malabsorbs and requires high energy, high fat diet. However, massive caveat to all of this is that we cannot yet predict the future. We need to be really, really cautious, in my opinion, and not cavalier about making assumptions, as I have posed here in an ideal scenario. I'm delighted that there are studies worldwide looking carefully to document any risks associated with drawing conventional treatments. And I think it's really important that we map these out and that we probably look for them more carefully in younger age groups also. It's imperative that we don't undo decades of progress with clinical care by being over cavalier in this age group. However, I think there are some concerns that postnatal treatment may never quite cut it. It's unlikely to fully work for every infant. We already see a variability in response in older patients, which is incompletely understood. We're likely to see this also in infants and children. Some of these children may not tolerate the drug or may have unacceptable side effects. Male infants will likely still have congenital bilateral absence of vas deferens, which is present antenatally. And perhaps it's more likely to envisage a scenario where disease is delayed or rendered milder rather than completely prevented by postnatal treatment. So the question gets put occasionally and is obviously inherent in the title of this talk: should we be starting these drugs antenatally? Well, there are some really compelling data in support of that from the G551D cystic fibrosis ferret. This paper looked at both in utero exposure and postnatal Ivor Captain administration to these ferrets. I'm going to focus here only on the data which came from the in utero exposure, but we can see benefits in multiple organs. Over on the far left, a reduction in meconium ileus. In the middle, maintenance of vas deferens patency, and on the far right, maintenance of normal pancreatic infrastructure. So certainly confirming work that has shown that these drugs can cross the placenta, they can get into the developing fetus, and that they can prevent some of the structural manifestations of CFTR deficiency. So far, there has been one also really supportive case report published here in JCF from New York, a woman with cystic fibrosis who was heterozygous for 508del, and her male partner who was known to be a heterozygote carrier of that mutation. They'd had unsuccessful attempts to conceive until 6 weeks after she started triple therapy. Now, improvements in fertility have been fairly well documented in women taking highly effective modulators, and I think we'll be collecting more of these data in the near future. This woman elected to remain on the drug despite its unknown safety. Following a concern at 20 weeks gestation of some echogenic bowel, this appearance had resolved by 32 weeks, and a healthy female infant was born almost at term. Her weight was above the 98th centile for age. The newborn screening blood test showed that her IRT was below the cutoff requiring further testing, but because of the family history, she went on to have genetic testing and was found to be homozygous for the F508del mutation. Despite this, her fecal elastase on several measures was above 500 micrograms per gram, confirming pancreatic sufficiency, and her sweat chloride at 5 weeks postnatally was 60 and 67, levels which are very rarely associated with this combination of mutations. So this case really supports the transplacental carriage of these drugs into the fetus, supports the concept that there can be some maintenance of organ health by antenatal exposure to these drugs. There was also a possibility that some of the immediate postnatal course was influenced by exposure to lower level of drugs in the breast milk. But I think there are a lot of challenges inherent in thinking about antenatal treatment outside the context of a small case such as that. The first is how we would confirm safety and how we would monitor this. The second is most of our families would not know about their fetal CF diagnosis in time for this treatment to start. Of course, this could be useful for families who already have a CF baby and have had antenatal diagnosis, but certainly in the UK, the vast majority of our parents don't know that they are carriers of the CFTR mutation until they have their first infant. We would have to try and figure out when to start and whether this is essential right from the beginning or could, for example, skip the first trimester. And then I think there are potential licensing and reimbursement issues to consider. Unlike the case I've talked about, most women carrying a CF fetus will not have cystic fibrosis themselves, so they will not necessarily be eligible to receive these drugs, and this may pose challenges moving forward. So to summarize, I think the future is extremely bright for today's newborns who are suitable for highly effective modulator therapy. It's essential we don't forget the 8 to 10% who are not on the basis of their mutation type. And it's encouraging that there's a very active preclinical and trials pipeline, but it will be some time before these young people are able to benefit from CFTR restoration if and when some of those treatments come to reality. It's imperative that we capture all the benefits and side effects of these treatments, particularly in young developing children. And we need to consider how care should be adapted as this learning increases, whether we establish different models of care, and whether or not we consider later or personalized introduction of symptom-directed therapies. In this regard, I think it's absolutely essential that we don't become complacent and that some of the perhaps very optimistic scenarios I've posed today are not assumed to be reality until we have those data collected. Thank you very much for your attention today.
All right, thank you all for joining us this afternoon. My name is Raksha Jain. I'm an adult pulmonologist at the University of Texas Southwestern in the United States, and I'm joined by my co-moderator, Paul McNally, who's a pediatric pulmonologist at the Royal College of Surgeons in Ireland, and he's been very generous in being willing to field the Slido questions. So I encourage the audience, please enter any additional questions you have, and also vote on the questions there so we know what your priorities are on what you would like answered. I'd very much like to thank our speakers, Amit Euler, Dave Nichols, Herman Beeler, JP Clancy, and Jane Davies for these excellent talks. I think it's been really exciting to see in the last couple of years where highly effective modulators have come, what we're learning, and where we can go in the future. So I'm just going to start this by going around and asking each speaker 1 or 2 questions that have been highlighted by the audience, and if we have time, we'll circle back with additional ones. So Amit, I'll start with you if that's okay. I think we've learned from your talk where we're going here, and that there's actually still potential to improve on what we've seen with current highly effective modulator therapies. An interesting question came about on if you have any thoughts, and we understand there may not be data yet, but any thoughts on what it would be like to combine ETI with the new ELOX drugs in those who may have a combination of an F508 and stop codon mutation?
Yeah, no, that's a great, thank you, Raksha and Paul again. That's a great question, obviously one that I really can't answer here today. With, right now we're looking at the role of Ivor Captain, but, truly having correctors and potentiators in addition to ELOX would be certainly a direction I would love to see, happen with the ELOX studies. But, the reason for going to Ivor Captain is there is, some thought that this would be, a good direction going based, with some potential data that suggests that might be helpful. So, there's obviously nothing published yet on this, and certainly nothing that we can really point to. But, I think it's really exciting. I think that is a great question, and I would be a proponent of that if we're able to pull something like that off. But not so easy to design something like that.
No, I agree. That would be a big challenge, but it's certainly an interesting question. Another question came about whether there is any data yet on those who have been potentially non-responders or maybe responded not as much as we would have liked to ETI, and whether any of those people are particularly being looked at in these new studies and their responsiveness to the newer CFTR modulator therapies.
Yeah, so in the upcoming, I think you mean in the upcoming phase 3 trial, looking at 121 along with deuterated ivacaftor and tizicaftor, and ETI. No, we're not looking at specific. It's not designed specifically at those patients, but certainly those are the patients that we are considering. At least that's what I'm thinking about when I think about additional modulators being available, because there are going to be. We all have those patients who either didn't tolerate or didn't have quite that response, and we're considering other studies to look at why that's the case. I mean, one dose doesn't fit all, and potentially, not all these modulators act the same on every single patient, as we already know. So definitely, that is an exciting potential benefit of getting other modulators onto the market if we were able to show that is that's what's going to happen with phase 3 studies.
Great, thank you. Dave, I might turn to you for a few questions here. One also came up about this group of patients that may be non-responders or did not respond as much as some of the others to ETI, and that is in the PROMISE study. Do you think that the data will allow you to take a better look at that group and even understand potentially the characteristics of those patients or gene modifiers in those patients to understand why they may not have had as much of a response to ETI as others?
Yeah, thanks, Raksha, and Paul too. Sure, the one purpose that wasn't mentioned in the PROMISE study is to develop a robust database and biorepository of samples that will be open for investigators who aren't in PROMISE from the beginning to apply to with their research ideas and to consider questions like that haven't been prospectively addressed. So that may be possible. I would say, though, that despite some modest correlation between FEV1 and sweat chloride, a lack of change per se in FEV1 did not prevent improvement in symptoms. And I think it's important to note, too, that coming in at a baseline FEV1 that was pretty high definitely didn't prevent people from going up further, and similarly on the lower end of FEV1. So the idea of non-responders in the ETI population, I think in our data set may be a fairly small group, depending on how you define that. But yes, there is an opportunity to look at some of that.
Terrific. I think in the same light, people were asking about whether side effects to ETI are being collected in the PROMISE study. I think there's been particular questions or concerns about mental health side effects with ETI and whether that will be captured in the PROMISE study.
Yeah, thanks. I agree. It's an important question. It is not a predefined or intentionally collected group, and we were intentional and fairly careful to not make this a safety study of an approved drug. We didn't think that was our role in doing so. Our symptom reporting is really focused on respiratory patient-reported outcomes and GI patient-reported outcomes that have been used for research. There is some general AE reporting and med use, but not any detailed reporting of side effects, and in particular, some of the questions that have emerged around mental health or rash or things like that.
Okay, terrific. Thank you, Dave. JP and Herman, I might turn to you next. People are asking a little bit about, I know you mentioned how people can go about utilizing the Cincinnati lab to send cells off. One question that came up is, how do they really identify people who might be eligible for that theratyping opportunity? Is there a place where the genes are listed? And I think along with that, if someone has already had mutations that have been tested, is that available for others to see so they don't have to go about sending the same type of genotype out to the lab to take a look? Could that be used for another person?
Well, first of all, thank you again for that question. And again, thanks for the opportunity to sort of highlight that work that's going on with that NRC. The first part was, I remember the second part for sure, that that I do know that their group has been able to take data from subjects with similar genotypes or same genotypes and apply them to other individuals. Often it's been within a family where one, for example, you might have two children who have the same genotype. That's very rare. And testing on one has led to approval for both. But I do think it's certainly worth reaching out to the center about their experience with different alleles or combinations of alleles, because you are correct. They, in theory, could provide sort of results from testing of someone who had similar genetics to another person with two rare mutations. So that's certainly available through their just directly through contact with them. The other piece about, is there a listing of the mutations that would be potentially amenable? And the short answer is there is not. It's the best way to go about this is typically to reach out to them specifically, and they will look at the sort of on a case-by-case basis to really evaluate whether the type of mutation is likely to have available protein. Some of these, like missense mutations, are pretty straightforward, and another one that's relatively straightforward are premature termination or stop mutations, which, unless they're at the very end of the gene, are unlikely to have protein just because of a process called nonsense-mediated decay, and that's required, just leads to very little available mRNA for transcription, for translation. And then the last questions, the tougher ones, is when you're starting to look at splice mutations, whether they could be what are called canonical or non-canonical splice. And some of those will have some full-length CFTR protein product, but it does tend to be pretty case by case. I think the reason that they've shied away from having a hard and fast line is simply because, one, each person has two alleles, so there's always a chance of one or the other might be responsive, and two, I think they want to be careful not to be to exclude someone. If someone, for example, saw an allele that would not respond, but perhaps their second one would, if that wasn't listed in the right way, perhaps that wouldn't be, that subject wouldn't be evaluated, if that makes sense. So the best thing to do is to reach out when you have rare genotypes, and they're happy to have a conversation about the rationale for sending cells and doing the testing.
Terrific, thank you. And Herman, this may be directed a little more to your talk. I think you mentioned this publication that you have coming out with some new variants that you all have tested. Do you have a sense of how many more people these new variants would cover?
Well, it's a good question. We think that we hopefully might be able to cover another 1% of people with cystic fibrosis with our in vitro theratyping study, in addition to the 2% that were approved at the end of 2020 by the FDA. The 2% of people with cystic fibrosis with rare mutations that haven't been covered before. So hopefully we will get to another 1%. I think that's what we think we can get to. I mean, there is a 7% of people that we can not target with these with in vitro theratyping because they need other, like RNA or DNA-based therapies or read-through therapies. But we hope that we can get to like a coverage of 93% of people with these effective modulator therapies.
Terrific, thank you. And Jane, I'll turn to you. I think you actually so far have the most popular question of the day. You showed us some really exciting data on the case report from New York of this mother who took a highly effective modulator therapy in her pregnancy with a carrier husband and was found to have this infant that had two F508 mutations and this exciting data. Do you envision, and I know this is somewhat just getting your opinion and view on this, but do you see a scenario in which a non-CF pregnant mother carrying a known CF fetus could be treated with highly effective modulator therapy to prevent disease in their newborn?
Hi, Raksha. Thank you. And thank you and Paul for giving me this rather intriguing title to talk to, which made me do a lot of thinking. Obviously, this is a topic that I've thought about quite a lot, and I alluded to it a little in one of the later slides in the deck. I find it difficult to imagine anything other than very, very small numbers of women that this would be applicable to, at least in my own country. We have very infrequent heterozygote carrier testing, which happens really on a family by family basis, because either parents have had a previous child affected or there's somebody else in their pretty close family. And so I think for this to be applicable to anything more than just that very, very small group of people, we might have to be in an environment where heterozygote carrier testing was more widespread. And of course, there are lots of examples of that for other diseases in countries where beta thalassemia is very high, for example. Carrier testing is the norm. But I'm not aware of whole countries where that is the norm at the moment. And therefore, I think most CF fetuses probably do occur in families who are unaware of their status. What I did wonder was whether or not any of the high-risk features, for example, echogenic bowel, or a family who for some other reason had CF detected on antenatal testing of some sort, whether that might be amenable to treatment. And I think I'd be quite interested in the views of those of you who don't come from nationally reimbursed healthcare systems, because I found it really quite difficult to think of a structure within which it would be easy to access drugs for a woman like that, at least in the National Health Service. And that might be something that is more possible in a system which is funded otherwise. I couldn't think of any other scenarios where a healthy woman carrying a diseased fetus was treated systemically with a drug purely so that it would cross the placenta. But it did get me thinking that it's perhaps not a very long way away from in utero surgical procedures, which are sometimes done, for example, with diaphragmatic hernias, and which really do prove life-saving, but which of course do pose a risk to the pregnant woman in the way that perhaps exposure to a drug would do also. So I think the bottom line is I really don't know. I find it a little difficult to imagine, but very interested in others' ideas. And I'm not surprised it's a popular question, because I think it is a bit futuristic and rather intriguing.
Yeah, no, I absolutely agree with you. And, I think not all people here in the United States are having carrier testing done either, though it's encouraged. And whether or not there could be any reimbursement or all the ramifications for treating a mother without CF with highly effective modulator therapy, I think, is yet to be known. So I don't know that we're in a different boat here in the United States. Anyone else is welcome to chime in, but I think despite our different payer system, we're probably in the same scenario. Terrific. There was some additional questions related to the data you showed on the improvement in exocrine pancreatic function, and whether or not you thought that adults, for example, should actually have fecal elastase testing done routinely after highly effective modulator therapy to see if we need to titrate their pancreatic enzymes.
So that's a good question. Personally, I'd say probably not at the moment. I mean, we haven't seen significant numbers of adults demonstrating restoration in pancreatic function. And certainly if we were to talk to our own dietitians, they would say they don't use fecal elastase as a guide to how much creon or other pancreatic enzyme to give somebody. They go on other measures. And there's not really a close relationship between the requirement for enzyme supplementation and the pancreatic fecal elastase threshold. So I don't think it's useful in that regard. I think it's more useful as a sort of binary on-off sort of decision-making. Has this person got sufficient restoration that they no longer need to be taking these enzymes? And I think that's sufficiently unlikely in adults, at least on the basis we've got at the moment, that I'm not sure that regular monitoring would go down very well with people. I mean, people are not, they don't find stool samples really popular. And for the yield, I think it may not be worth it. But I think what's important is that we have it front of mind, and that when we're history taking or when we're examining patients, we're thinking about, is this a possible scenario in which it would be useful for us to have a look at fecal elastase and see whether, in fact, to our surprise, this patient has converted. I mean, it's not very long ago we thought pancreatic insufficiency was irreversible even in infants. And now we know that's not. So we may be going to find that there is also some conversion in adults. But I think we should probably do it on a case-by-case basis rather than completely across the board.
Terrific, thank you. Dave, I think I'll circle back to you because there's several more questions about the PROMISE study. Some of those are about the microbiology data you showed and whether there's going to be data about non-tuberculous mycobacterium, particularly M. abscessus, as well as Burkholderia cepacia, and whether people will learn more about whether those organisms are going to change after ETI therapy is introduced.
Yeah, it's a great question. So just the inherent limitations in any study, looking at very rare infections are relevant to PROMISE as well. We are not doing mycobacterial cultures. As was alluded to earlier, sputum is an increasingly precious commodity, and so we've maximized the use of those for what we think may be most efficient. There are data on Burkholderia cepacia. The numbers were relatively small, as I recall, 10 to 15 people who grew that at baseline. And we'll also go back to the registry data and try to identify those who may have been intermittently positive for that, but not at their single baseline visit. Again, we'll point people toward Sarah Morgan's talk, where I think those data will be shared as well. And there was a poster last year showing, but in essence, reductions in Burkholderia cepacia positivity were similar to those seen in Stenotrophomonas and Pseudomonas. Impressive reductions, early on, but those need continued follow-up to really understand, is that sustained reduction or perhaps even elimination if we really scrutinize those samples. I think you're on mute, Raksha, sorry.
Thank you. Another question came in about the correlation data you showed with sweat testing and lung function, where you showed that the cohort actually did demonstrate a correlation between FEV1 and sweat in this PROMISE data. Did you take a look at whether, if you limit this group to those who had a lung function between 40 and 90%, to remove those with higher chances of having a floor and ceiling effect, did that change your results at all?
It's another great question. This is always helpful to hear these. So we have not looked at that. I'm sure we could go back and do that. There were really three reasons that came to mind for us as a team as to why we were able to see that correlation, albeit modest. One, probably most important, the effect size, the large effect of ETI on sweat chloride and FEV. But 2 and 3 are relevant here. A fairly large sample size we were able to enroll in the study, and then the breadth or heterogeneity of response, because we enrolled a real-world cohort with a lot of variability in baseline modulator use and starting points. So I'm concerned that if we were to limit to 40 to 90, we would reduce our sample size and that heterogeneity or spread some, and that could be lost. But you can't know until you look, so we should definitely go assess.
Great, thank you. Amit, I'm going to come back to you with a question. It was asked about, as these studies are being designed with highly effective modulator therapies, now that people have ETI, do you foresee a challenge with these new clinical trials and enrollment, specifically the AbbVie ones? Do you think we're going to be able to enroll in these studies in a robust enough fashion to get good data and thoughtful data on new modulators?
Yeah, no, that's a great question, and thanks. I think that, the reason why the AbbVie studies are being performed overseas and in places where triple combination therapies aren't available is, the reason for that, and, for the ethical issues that would come up if we tried to enroll patients, here where ETI is more readily available. Then the designs are going to be similar to the 121 phase 3 study, where we're comparing, these, head to head, the the modulators together. So I can't, so I do see, we'll wait to see how enrollment is going to be in these 1-to-1 studies. Although I think that from discussing, locally, that we seem to think that we're going to have enough people enrolling again for these trials, as people with CF are always interested in trying to push the envelope. But this is we'll wait to see what happens. And with and I thought maybe you were going to ask about with ELOX drugs, whether adding these, sort of adding them would be, designing it would be, I think, feasible to do in the future, pending, the results of the phase 2. But I think that we're going to continue to have these issues but sadly, there are going to be areas of the world where ETI is not going to be available for some time, it seems. And, perhaps that's where all these modulator studies are going to take place.
Got it. Another question came in about the design of some of these studies, and specifically the design of the Vertex phase 2 121-101 study. But do you recall the duration of the washout period in this study as people are thinking about washout studies?
Yeah, I believe it was, and God, I'm gonna hate myself. I think it was 4 weeks of the washout, but I'm now guessing it wasn't, now that I said that. But no, I think that is, important to have those washout period, for patients enrolling. But I can't recall exactly what it was now.
That's fine, no problem. And if you happen to come up with it, you're welcome to chime in if you want to look in the meantime. And Jane, I might come back to you as another pregnancy question has come in, asking about whether there is yet any data available or studies on CFTR modulators, specifically in pregnancy and looking at safety. And the other being, how concerned should we be about cataract formation or vision impairment in infants born to mothers who continue to take Trikafta?
So I think that's a very good question about, we need to learn more about safety and outcomes in pregnancy. And yes, very pleased that colleagues, I think globally, are trying to collect multiple case series of these. I think that's probably the best evidence base, rather than there being any sort of prospective study envisaged. But Jen Taylor-Cowser, I know, is leading on that and is really very keen to gather as much information on both maternal and fetal outcomes as possible. I think the ophthalmologic concerns, of course, will form a part of that. And one of the safety aspects, of course, is that this signal, when it was picked up in the preclinical toxicology models, was something that was seen related to both prenatal and immediate neonatal exposure, as opposed to in older animal offspring. And so it's something that certainly within pediatric care, we've been very, very keen to look out for in infants starting Ivor Captain early in life. The numbers being reported, I think, are still gratifyingly extremely low. And certainly to my experience, the vast majority of changes that are picked up don't seem to be visually clinically important. But I have no idea. In fact, I would say probably the eye is the organ of the body about which I know least. I probably missed that lecture in medical school or something. I couldn't begin to tell you about the risks of an in utero fetal exposure for something like this. But, you would think that it's going to be substantially greater than any postnatal exposure. So it would certainly be something that would need to be looked out for. I haven't seen it reported in the few cases that have been, I think, reported to date in the offspring of mother who's been taking ETI. But obviously those numbers are still small. So we need to be really circumspect about that.
Thank you. And I might just take a second to plug, there is just begun in the United States, a study called the Mayflower Study, which I think you alluded to, that Jen Taylor and others are leading, and this is sponsored by the CF Foundation and being managed by the TDN. But this will be one of the first prospective studies of women who become pregnant, who are both either on ETI or not, and following them forward. And amongst the data collection will be collection of infant data that is going to happen naturally, but it's being encouraged that women who take highly effective modulators have their infants checked for eye impact. And so we may be able to collect some data if pediatricians and mothers choose to have that assessed in the baby. So in a year or two here, we may have a little more data to answer that question, I hope.
That's great that that's happening. Fantastic.
Good.
Raksha, just to get back, it is 4 weeks was the washout period before and after, but I just wasn't sure, what the question was asking, whether that is the appropriate amount of time or.
I think they were just curious of how it was designed and how much time. So I think 4 weeks is exactly what they were looking for. So thank you, Amit. JP and Herman, I might come back to you. There is a question about with the data that has been collected thus far on determining whether people are responsive to ETI based on these cells, is there any data collected on how many of these cases actually translated to someone being able to get the ETI approved or a highly effective modulator approved for use after this data was submitted to insurance companies or payers?
Yeah, first of all, happy to weigh in a little bit about that. It looks like, again, if you just take a look at all comers so far, they've had about 45% of people who have submitted cells for basically modulator testing have demonstrated what we consider a response, and the response definition that they're using is similar to what was described by Herman, achieving 10% of wild type levels. Truth is, we don't know for sure how meaningful that number is. It is the same one used by the FDA and thinking about label expansion in FRT cells, so there is some precedent there, but it's still a little unclear whether a 10% improvement in the in vitro system or achieving 10% of wild type levels in the in vitro system really translates into some measurable clinical benefit. But of those roughly 45% of patients who meet that threshold, about 75% have actually been able to get access to drugs based on the information alone. I think an important question that is not part of the study, and it's simply because there's just so many different sites that are providing cells, is how have the patients done? have they actually demonstrated benefits? And the cleanest way we think to really be able to address that is to be able to, in theory, re-enroll those patients in the CHECK-SC study. And in that study, what that is doing is looking again at the value of sweat chloride while on drug and how it relates to clinical outcomes. Could be looking at change from their diagnostic sweat chloride or else a threshold level to determine what exactly that change or threshold level of sweat chloride predicts in terms of clinical outcomes. But the cleanest way to do it would be through the CHECK-SC study, which has numerous sites across the country.
Terrific, thank you. Dave, I'm going to come back to you with a question or two. And one of them is in your talk, you mentioned how some of our traditional endpoints are vanishing a little bit because we're hitting a ceiling effect on some of them. And it was, we received a bit of a broad question about how do we go forward from here and looking at these important endpoints, and maybe particularly in children where the ceiling effect may be even more prominent.
Yeah, that's $1 million question. I don't think I would be so bold to use the term vanishing. I would get in trouble, my colleagues, for that. But the truth is, I think we do need to be open-minded about alternative endpoints in certain studies. Dr. Davies alluded to LCI. I think that's an important one to consider, especially in younger populations. Perhaps alternative microbiology measurements if sputum is difficult to obtain. We have recently done some work about home collection of sputum, asking individuals with CF and their families in the US on ETI if they ever produce sputum at home and their willingness to bring that in for research or mail it in. And the numbers were surprisingly good. So that may be an alternative outcome in that regard. The symptom reporting, I think, is the one where we may face the greatest challenge if these data are durable. Looking long-term follow-up studies sponsored by Vertex and real-world studies, if reported respiratory domain scores remain at or near the top, then that is a little harder, I think, to try to deal with. And so we may need to re-envision and better understand what symptoms persist, what people with CF are still being bothered by, and go from there. The CFQR CRIS is an alternative PRO focused on respiratory symptoms, and that hasn't been tested thoroughly yet, and so worth more consideration, I think, in this population.
Raksha, I'm just going to jump in because one of the questions you asked me before was about study design in the future, and just sort of bouncing off that, LCI we're using with children and not with adults because we you may have alluded to this before, because of the long washout period and not so feasible to use in adults with advanced disease, but with adults who now with seemingly normal lung function in some cases with less disease, perhaps LCI in adults might be more feasible in the future, looking at these like lung functions above 90%.
Terrific. Is that part of any of the current design of the trials that you mentioned?
No.
Not yet.
Yeah,
okay.
But something to consider.
Okay, great. Amit, while you chimed in, I might ask you another question or two that came in for you about some of the side effects of the medications known. So for example, one of the questions was with the newer 121 triple combination studies, do we know anything yet about side effects? And some of this data may not be available yet, but do we see a decrease in the impact on liver function testing with these newer therapies relative to what we've seen with ETI and other modulators?
Yeah, no, I think that important to note that in the phase 2, there wasn't matching, it wasn't one-to-one looking at comparing to Trikafta or to ETI, sorry. So I don't think we can say that. And in the phase 2, just nothing different than what you'd expect from other modulator studies is as far as I can say. But so yeah, I don't think that we can make I can answer that question fully.
Got it. Thanks. Dave, I'm going to come back to another PROMISE study question, if that's all right. You commented a little bit on adherence to therapy that you saw in this study and some of the measures you use. People also asked about compliance to pancreatic enzyme replacement therapy and whether any of that is being collected, particularly in the GI sub-study of the PROMISE trial.
Thanks. So it's not, I'll answer that directly. It's not a focused question. We are collecting all ConMed use though throughout. Perhaps there'll be some information around dosing. I'd have to be sure that we're collecting that level of scrutiny. I think we are, but we did specifically ask at each study visit around 4 medications of interest or of maybe greater interest, including hypertonic saline, Dornase, and inhaled antibiotics. And we encouraged individuals in this study to try to understand the effect of ETI to maintain their other regimen at a stable use pattern through the first 6 months, so which is what we've reported on here. So for the most part, that did happen. There was some reduction in inhaled therapies, and the largest was seen in inhaled antibiotics. And just a quick shameless plug, this has been published or accepted for publication. So all those data will be available to look at in more depth here in the next couple of weeks coming out. There was about a 30% reduction in reported use of inhaled antibiotics at 6 months compared with baseline. We have not yet looked to see if that associated with changes in their microculture status.
Okay, great. Along the same lines, I think people asked a little bit more about adherence, and given, I think, the general theme that many have seen of people with CF starting to limit their use of traditional airway clearance therapies, I think people are expressing that they've had problems advocating for patients to maintain these other therapies. Is there, I know there's, maybe I'll have you comment on the Simplify study because I don't know that everybody knows this, but I think the question is there a faster way to address this issue and give people data to understand how to advise patients?
Yeah, thanks, Raksha. For those who may not know, there is a study called Simplify, led by Nicole Hamblett, Alex Gifford, and I, were looking at short-term changes in lung function, including FEV1, LCI, and then a smaller outcome measure looking at mucociliary clearance, led by Scott Donaldson, testing what happens when people on ETI are doing really well, stop versus do not stop, hypertonic saline or Dornase alpha. So those data are expected to be available by this time next year, perhaps a little sooner. It's a large study testing for non-inferiority, trying to really scrutinize whether or not there is a cost to lung function across these outcome measures, at least in the short term. So that will add to other studies, several other studies trying to understand this. And I just want to echo what was said earlier, that I think it's important that we are very careful about this and really try to understand what costs there may be in stopping other symptomatic therapies. I don't think we know that yet, and it may be hard to feel that. People, feel pretty good on these drugs, and I'm concerned we could lose out on some other health benefits if we stop too abruptly.
Thank you. That's a great point. Jane, I wanted to come back to you on a question. I know with your expertise on LCI, a question came up if you could comment potentially, particularly on the EMA view of LCI as an efficacy outcome measure, particularly as we move forward with some of these pediatric trials and other trials of modulators. Is that a tool that is being more widely accepted in the UK and other places?
So thanks a lot. And yes, I've been very pleased by actually the approach from regulatory towards LCI. The studies in which it's been approved as the primary outcome so far have been studies of drugs which have already had a license in a different population, and we're looking to extend that usually down to a younger age group. We haven't yet approached the EMA with a study for a first-in-class license using LCI, and I think at the moment that might not be met well. I know we've talked to them as part of groups on behalf of teams trying to develop other outcome measures, and the data set which they would wish to see in order to approve a primary outcome for a first registration would be really very, very extensive, and concerns me somewhat because there was a lot of talk at certainly one meeting I was at about them wanting to really see correlations with survival data. I think we've got to a situation which is very, very fortunate in CF now, where the vast majority of things are not going to be, we're not going to be able to gather data on their long-term impacts on survival because the survival is now so much longer than it has been in the past, and that makes that an extremely big challenge. But this is something that we've got aspirations to do, to open up a conversation with regulatory about this. Slightly gone on hold during the pandemic for understandable reasons. But I think that nevertheless, although it's going to be really, really useful, FEV1, which is quite well regarded by them and is well understood, is likely, at least for the foreseeable future, to remain useful in the adult population. And that's where drugs are mainly going to be tested, at least initially. I could foresee in a few decades' time, when we transition very, very healthy children into adult services, that we might have the need for something that's a lot more sensitive, even in much older adults. And so I think it's still important that we're working towards that aim, and alongside other perhaps new imaging techniques, we'll have to try and think about ways in which we can achieve that.
Great, thank you. And I know we're running short on time. I might just put one more question in there for Amit, and this is sort of a broader question, but someone asked when comparing VX-121 and elexacaftor, can you explain, I know you explained the new once daily dosing potential of this therapy, but outside of that, are there clear potential benefits of VX-121 over elexacaftor in particular?
Yeah, and once again, we're going to be hopefully presenting this data in a manuscript form soon. And also the phase 2 study wasn't done head-to-head with Trikafta, so I think we really can't say if it's better or not. I know that in vitro data suggested that there was an improvement in with using a using chamber, looking at the and CFTR function, that there was a potential improvement seen in vitro, suggesting that that we proceed looking at 121. But beyond that, I think that we can't say too much more until we do the head-to-head trial, which is coming up with the phase 3, and so that will hopefully answer that question, the next year or so.
Great. Okay, so thanks a million for that, Amit, and we're just close to wrap-up time now. So on behalf of myself and Raksha, we'd like to thank the organizers for allowing us to put this session together. I think people will agree it was a really fantastic and insightful session from world leaders. And thanks so much to all the speakers for their great talks and also for their attention in a marathon question and answer session, which I think was really informative. So thanks a million again to the audience as well for participating, and enjoy the rest of the conference. And just a reminder that the odd number poster session is on in about 10 minutes, and in an hour at 2 p.m. Eastern, I think, the first plenary, and Michelle Prickett will be delivering that, and it's about meeting the moment, lessons for changing. I'll read the rest of that. Lessons for a changing world. So enjoy that and enjoy the rest of the conference, and thanks a million, everybody, for your attention.